Hi, my name's Lisa Butt. I work at the University of Exeter and I'm studying plant diseases. Plant disease is really important, both from an economic point of view and also from an ecological conservationist point of view. It's estimated that around 10 to 20% of all crops globally are lost every year due to plant disease. It's obviously a huge problem. It can cause economic instability, but also, perhaps more importantly, it can also result in serious food shortages and also, in extreme cases, famine. Rice is a globally important food crop, and many people rely on it as their main source of food. This here is a rice plant, or it's several rice plants actually in the same pot. These have been growing in my house, so normally they would grow in a growth chamber that has perfect humidity and regimented watering times, and so they're looking a little bit worse for wear, but I think they're doing okay. They're still pretty green, so that's I'm considering that a plus. And rice can be infected by fungi, by bacteria, and by viruses, just like people can. I'll put that down over there. So scientists have been trying to find ways to combat these diseases, and in the past, chemical pesticides have been used to control diseases. But there are some problems associated with these pesticides. First of all, is that the microorganisms, that is the bacteria or fungi or whatever it is you're looking at, that are causing the disease, they can become resistant to the pesticides, much like human diseases that can develop resistance to antibiotics. This means that the pesticides are no longer as effective as they were, and that means they're not as good for controlling outbreaks. Another issue with the pesticides is that they can also have a quite serious effect on bees and other insects, which in turn has an effect on other plants, other animals, birds, all in the ecosystem. This can have a real negative impact on the environment and can last for quite a long time. So reduction of these pesticides is something that a lot of researchers are looking into, and we're looking for alternative methods to control the disease. One potential idea about controlling disease is the use of bacteriophage. So bacteriophage are viruses that can only infect bacteria, and they tend to infect very specific bacteria. So they can only infect one species or a couple of related species more often than not. So these can be used to control bacterial diseases really well without having an effect on the plant or on the other microorganisms that exist within the plant or on the plant that can actually be beneficial to its growth. So these are really good, and I've recently studied bacteriophages myself, so this is something that I potentially come back to as an area of study, which would be quite interesting. Another approach is the introduction of another species of microorganism to compete with the disease-causing one. So this can do things like slow the growth of the disease itself, and as a result, that can allow the plant's immune system, because plants have immune systems just like people do, the plant immune system can kick in, and then the plant can stop the spread of the disease itself. So these interactions between different microorganisms is something that a lot of researchers are very interested in. Something that's been studied in humans already quite a lot, so dual infections and looking at the microbiome and stuff like that is already something that's been studied a lot, and people are starting to develop it in plants now. So my current project is looking at two different species of bacteria that can infect rice plants. So I've done some quite terrible cartoons, so I can only apologise now for the drawings. So these are my two bacteria. That's Burkholderia glume and Xanthomonas oryzae. And both of these bacteria can cause quite devastating diseases in rice by themselves. What I want to look at is how they interact with each other. The microorganisms can interact with each other in a variety of different ways. the first that we could potentially look at is cooperation. So cooperation is when one species of bacteria produces something that is beneficial to both species. So here's another rubbish drawing. Here we go. That is an illustration of cooperation. I've got my finger over it there. So you can see one bacteria there is handing, it looks like a book, it could be anything, over to the other species. And this allows them both to either grow quicker or to evade the immune system, or to both just grow more efficiently. And it just means that the disease can progress quicker, or it's harder to eradicate from the area. So cooperation is one potential way for the two different species to interact. Another way is antagonism. So antagonism is another picture. There we go, is when one species produces something that can either kill the other species or it can just stop its growth, allowing the first species to then grow as much as it wants, and stopping the other one from carrying on at all. So looking at these interactions, we can then look at the best way of controlling the disease. And that is something that I want to look at. I want to look at how these two different species interact inside the plant. So I'm a microbiologist, so what I do is I work in a microbiology lab, and we grow up bacteria and we use it to infect plants, and then we look at the infections and see how long it takes for the disease to hold. We look at how the two bacteria interact with each other, whether introducing one will halt disease or make disease a lot worse. And these are things that the plants, dual infections are things that plants are often subjected to in the field. So it's something that is worth looking at, and then that would lead us on to potentially looking at bigger experiments, or a different lab looking at bigger experiments. So that is my current project, and I'm hoping that I get some interesting results from it. And that's it. So I really enjoy working in a micro lab. There's always different experiments to do. It's always interesting, and you quite often find that your results aren't what you're expecting. So there's always new things to learn, and it never ever gets boring. And then we also have to be very careful. So once we finish the experiments, we have to make sure that everything is controlled and destroyed before it goes away from the lab. So we're very careful about how we remove everything from the lab. So that's part of being a microbiologist. And yeah, so that's what my research is about. Hopefully some people will be interested in looking into it further. And if anybody has any questions, then I'll be delighted to answer them. Thank you very much. Bye.